Mickey, can we start or are we waiting? בשירות של המערכת.
מצוין, שירות מעולה.
Okay, can we start?
Can you start on 2 minutes before you...
Okay, I need 2 hours.
Hey, Kurt, do you want us to set the clock?
No.
It's okay.
You have 15 minutes, 2 minutes before.
Okay. We'll manage.
Okay.
Okay, so this is a paper from Science 2003 by Fiorillo, Tobler, and Schultz, and it's called Discrete Coding of Reward Probability and Uncertainty by Dopamine Neurons. And it's a pretty straightforward paper, and we don't have much time, so I'll focus on the results. I'll just go through the methods quickly, and then I'll tell you the more interesting results that they have. And if we have time at the end, we can discuss the implications of what they show or the interpretations they give, or we can criticize the paper together, whatever you prefer. So this is electrophysiology in monkeys. So it's n equals 2 macaque monkeys, and they record while they're doing this task, which is just classical conditioning. This is a cartoon I made because they don't really show how they built the experiment, what kind of images they show, but they pair an image to a probability for reward, and it's shown for 2 seconds, and after that the monkey either gets a reward of some juice or doesn't get a reward. And overall, across many trials, each picture corresponds to some probability of getting a reward. So for example, this picture overall across all the trials, in 25% of the trials the monkey would get a reward, and in the rest he wouldn't. And the monkeys can learn this, and they did tons of tasks. It's about between 100 and 200 tasks a day, 5 days per week for 5 weeks. So it's a lot of trials. And the monkeys can learn it. This just shows the amount of licking that the monkeys show after the image is shown and before the reward is received or not received, depending on probability. And it's as a function of the probability, so you can see that this increases when the probability increases. And this will be across all the figures. The black dot is monkey A and the white dot is monkey B. So just keep that in mind. So it's not, it's not perfect or anything, but you can see that they lick more because they anticipate getting a reward in the higher probabilities. And they recorded single unit recordings from the ventral midbrain, which is, as far as I could understand, just a general term for the area that has most of the dopaminergic neurons, the substantia nigra, VTA. And here they show, this is the only figure that they show where they record from. And they co-localize the recording sites, which are all the black dots, with antibody staining for tyrosine hydroxylase, which is the precursor for dopamine. And since this doesn't, this wasn't clear for me, so I added this from just a recent paper, which does the same kind of staining. So you can maybe kind of see the connection between this black staining here for the dopamine or the tyrosine hydroxylase and the shapes that they show here. So this shows that they were recording in areas that have lots of dopamine neurons. And since they go in blindly, they identify the dopamine cells using the electrical properties. So they know that they are recording from a dopamine neuron, or they think they know. And then they co-localize it with the dopamine precursor. So methods and yeah.
So all the dots outside this region.
Yeah. So they are either dopamine cells in areas that don't have a lot of dopamine and or tyrosine hydroxylase and they weren't labeled, or they are not dopamine neurons.
Why is single unit important? It's multi-unit important, and they just in the algorithm they try to find.
I think they look to get like single responses. So they try and localize one neuron. But basically it's just extracellular recording. But they call it single.
It's extracellular?
Yeah. Okay. So they got two interesting results. The first is what they call a phasic response, which is basically a response that's centered around a specific time window. So there's a response and it decays. And it's centered around two places, and one of them is where the reward is either received or not received. At that time point, there is a response, and the other is when the conditioned stimulus is shown. So these are graphs of the median response as a function of the probability, and you can see that in the rewarded trial, the response of these dopaminergic cells decreases as the probability increases. So when the monkeys do not expect a reward, you can see a high response. And when the probability increases, this response decreases. And this is for the conditioned stimuli. It's the opposite. So when they see an image that tells them, so to speak, that there is no reward coming, you don't see a response. But when they see an image that tells them that a response is coming, there's a reaction. And this is just a response of a single cell, probably the best example, from monkey A. And this is when the conditioned stimulus appears, and this is where they either get a reward or don't. And you can see that, and these are the probabilities, so you can see that this response increases with probability, and this response decreases. So what they suggest is that this is some sort of prediction error coding, that the dopamine neurons code prediction error, which is the discrepancy between expected probability for reward and the actual outcome. And unlike the nice single cell that responded like to all of them, not all cells responded to all probabilities, but it's always monotonic. So the response for a higher probability is always lower at the time of the reward. And this kind of result was shown before. They're not the first guys, people to show it, or maybe they showed it before in a different paper, I don't remember. But this was shown, but it wasn't shown across the whole range of probabilities. So most people did either p equals 0 or p equals 1 and showed that this kind of thing happens, but they did it across the probabilities. And that allowed them to discover a new kind of response that wasn't described before. And this is a sustained response. And again, this is a single cell. And you can see that once the conditioned stimulus, the image is shown, there is some kind of activation which continues all the way to when the reward is supposed to come. And it increases towards the reward. But what's interesting is that it doesn't happen when the probability is deterministic, either at p equals 0 or p equals 1. And this is the population response, and you can see it more clearly, that it doesn't really happen here or here, and it's maximal around 0.5. And this is what it looks like for the whole data for both monkeys. So it's maximal at 0.5, and it decreases on both sides. So this is something that's very similar to other functions that code for uncertainty. This is Shannon entropy, and this is the standard deviation of a random variable, Bernoulli random variable. And they both have a maximum at the maximum uncertainty, which is p equals half, and they decrease on the other sides, and this looks kind of similar. And also the fact that this response increases towards the time of the expected reward, which is also the time that the monkey is most uncertain about what's going to happen, led the researchers to conclude that this is coding for uncertainty. So this is basically what I said. Some dopaminergic cells show a slow, more sustained activation, which increase towards expected reward, and it's highest for 0.5, and doesn't happen at 1 or 0. And previously, this wasn't described. They say either because people didn't look at the intermediate probabilities where this happens, and if you look only at 0 and 1, you won't get this. or because it's not a typical response for the dopaminergic cells, people thought, okay, so this is not a dopaminergic cell, and they just ignored it. And what's interesting is this only happens in motivationally significant circumstances. So if they pair an image with a probability of some other image appearing, which has no rewarding value to the monkey, then they don't see this sustained activation. So the sustained activation only happens when there's something that's related to uncertainty about reward and not just uncertainty in general. One last thing that's interesting is that they try to see if these are two different populations. Maybe one of them is really dopaminergic neurons and the other population is something else, or maybe it's two populations within the dopaminergic neurons. So they tried to correlate the maximal responses on both the phasic activation and the sustained activation. So this is phasic activation at p equals 0, which is always the highest, and the sustained activation at p equals half, which is highest. And you see that there's no correlation here. In fact, a lot of cells didn't even show much of a response to either. But this led them to conclude that this is actually one population that can code two different things. And I'll summarize. So it's two distinct response types in dopamine neurons that code two different parameters about the world. The phasic response corresponds to reward probability or prediction error, and the sustained response corresponds to reward uncertainty. And they both come from one population. Basically, that sums up the paper. How much time do we have?
2 minutes.
So I can say one minute about each of these kind of things to talk about here. The first is whether reward probability and prediction error, which they use really like interchangeably, is that the same thing? What is the phasic response coding? Because if you remember, it codes. You really see how it decreases with probability, but it increases for the conditioned stimulus. So you could think that it also just codes reward. And when you have an image that predicts a reward at a very high probability, you get the response as if you got the reward right when the image is shown. So you don't see it when the reward is happening. And that's consistent if you look. So when you don't expect the reward and you get it, you see a response at the reward. And when you expect the reward, you get the response right at the expectation point, and that fits models of CD learning, which we talked about in tissue view. So I don't know if their interpretation is exactly, if it has to be true. I'll skip the goal of learning, but they have an idea why we need more than one parameter, why uncertainty is useful. And this is related to this. Why use dopamine to code for errors or for uncertainty when we all think, at least I think, of dopamine as some kind of positive reinforcer, something pleasant, I don't know, drugs that get us, that make us have more dopamine are pleasant. So they have a theory that uncertainty is actually rewarding in itself, and if you think of gambling, then that makes sense in a way. People can get addicted to gambling, and people can get a pleasant feeling from gambling. So they say that unlike in the casino where the probabilities are always fixed against you, and gambling usually leads to losing, in real life, gambling and risk taking is often beneficial and might be a good strategy. So basically that's it. Questions?
Quick question. Someone has to get to the
Yeah, if you don't, I can keep talking.
Iman. Come on.
It's early. Maybe she has it. Well, everything
So,
else.
Okay,
how
yes.
many
Nobody
minutes?
wants to
Yes,
argue about the
I have
result.
a question.
Yeah.
Who's next?
Can I have a question?
No, yeah, go. Okay, so let's
In the first slide, way before, yes, where you describe the one after the probability.
Yeah.
When did they give a reward at the zero probability condition? Because after they gave the reward, it was at zero probability.
Yeah, so that's after like all the thousands of trials.
Mm-hmm.
So they do this. They don't say exactly how many trials they get actually the a suppression of the neural response. Um, but again, they have some missing data. Some of the, if you noticed in some of the graphs, there are missing data points. Like I'm not sure what's going on here. Um, and some of this is, and they're actually honest about it in the paper. They say that, for example, in the sustained response, they did, at the beginning they didn't think that it was dopamine neurons. So in some of these trials, they didn't do the whole experiment because they thought, oh, it's not a dopamine neuron. They just ignored it, so they don't have the data. So it's, yeah, the data is incomplete and it's not really clear. But my guess is that after many trials, the monkey thinks it's zero, and then they do.
Doesn't it change the result? I mean, doesn't it change the probability encoded in the?
So the first time it's okay, right? Because it's just surprise.
Only the first time.
Yeah. So I don't know how many times they did it, but I mean, if you do 10,000 trials and 3 of them is you get a reward. So yeah, it's not zero, but it's very close to zero.
How many cells do they have?
It's about 200. But not all 200 responded to both. So here, I think it's about 180 cells, and for the sustained response, it's between 5% and 30% of that responded, depending on the probability. And
Both in BTA and attention, right?
they don't differentiate; they call everything ventral midbrain.
But it's extracellular, so how can you know exactly the number of cells you don't record kind of cells?
Yeah, but I think you can see when you're recording just a single response, right?
It's very dependent on the electrode. If you use a very high impedance electrode, you can actually go.
You record for many cells together.
Right, but
No,
if
no,
you are
but
very close to one cell, you will
You have
record.
to see that you're very close to the cell.
Yeah, but you can see it on the recording. You can see by the size of the spike, and usually what you do in a condition like that, technically, what you will see is like very, very big spikes that all looks very similar to each other, and then many small spikes that are very different from each other. So if you just throw all of them out, you're left only with one cell spike. So it depends whether you try to maximize the number of neurons you record from, and then you would actually try to be very close to one neuron. And then you have to do spike sorting and all kind of things like that, or you just say, I don't care, I just want one at a time. Just do it many times.
So that's what they did.
I want to highlight a few things about this paper, although it's kind of completely outside my scope of expertise. First, this is like a very big paper in the sense that this is what?
Influential paper, not big.
Influential, and it's a major finding. I mean, the fact that you have like prediction error coding of neurons is very significant, and it made a lot of noise. And theorists actually, obviously, liked this paper a lot, and made a lot of work after it. But from my point of view, as you know, I want to highlight other things that are like what kind of the chupchik of the kumkum. From the way I look at things like that. First of all, you notice that they treat like a whole brain area as one thing. Like there are
It's not even one brain area.
It isn't two. I mean, there are millions of neurons there, and basically they're all doing the same thing, which is kind of very, very strange in my point of view. I mean, some people look at that this way, like basically the whole substantia nigra is doing one thing, which I find kind of surprising. It might be true, but I think it might be a matter of resolution. So far we are looking at a very, very crude task and a very crude resolution. So there might be much more going on there that we don't pay attention. Second thing, that no one, as far as I know, has ever looked at how is that being computed at all. I mean, it's a very complicated computation, right? You have to have like the expectation, and you have to have the result, and then you have to code for the difference. And I don't know of anyone that knows where one input comes from and when the other one is coming from, and how do you compare. Zero. So this is like a whole open field gyroscopes. You have the whole basal ganglia cortex loop, and then the subclavian ganglia is just giving you the arrow. But how is that? This is just a magic. It's like the arrow is coming from God. No one knows how that is being computed. So I think this is very important.
What's the approach to compute this?
What?
What's the approach? How you can
Well, you
really
need to
start?
know where the input is coming from to these areas and what type of input. Like sustained activity, for example, is usually typically correlated with prefrontal cortex activity. This is like short-term memory and things like that. So I don't know if anyone did like dual recording from prefrontal and subclavian ganglia and look for correlation, or did retrograde labeling from subclavian ganglia to prefrontal cortex. I don't know if any of these. And the last thing that I want to highlight is dopamine. So you already know the dopamine encodes for reward and everything, but dopamine, what is dopamine? The neurotransmitter, everybody knows. But what is it doing? What is it binds to? It's like activating glutamate receptors, GABAergic receptors.
Dopamine receptor. So it can
Dopamine
either
receptors,
excite or inhibit.
so dopamine receptors, but dopamine receptors are decoupled proteins. They are not actually ion channels. Okay, so you have to think about all these things in a very different way. So when a neuron is spiking here and the postsynaptic neuron receives dopamine now, first of all, it's not going to one synapse. It's like pods. It's not like dopamine synapses are not. It's not like a typical synapse. So the presynaptic neuron releases dopamine, but there's no postsynaptic side only with dopamine receptors. Dopamine receptors are all over the dendrite or the soma or the axon, and they're all kind of being activated. And secondly, they're not kind of opening ion channels. They're starting all kinds of processes in the neuron, getting cyclic AMP higher levels or lower levels, and this starts all kinds of other things, modulate ion channels. So it's not obvious to read out what that thing is actually doing. It's true that you have D1 and D2 receptors, but what it actually does to the neuron is a completely different story, and this has to be kind of taken into account. Right? Okay, Gabi.
הגעתי לבד מה מדוקטורים. אני מצטער. שנייה, ממש דחוף. רק תהיו לי קודם. Okay. I hope I'm a bit coughing, so maybe okay. Okay. So actually, this paper is heavily based on Gad's paper, so he planned it this way. So he starts in. I'll give an alternative explanation of these results. And so instead of uncertainty, as we'll see, they propose a different mechanism for explaining the same results. All right, so some of you, like Gal mentioned, learned in TV learning, but for me it was
Kindergarten. Kindergarten.
Yeah, okay. Kinda, it's the same. So in a nutshell, so in standard learning or supervised learning, the agent learns only by actually observing the value. So we have some prediction, and he observed the specific value, and only when the observation is available, then we adjust what we thought to what actually is, and then update our model of the world, and then go on. In TD learning, we're doing something else. We're kind of doing some kind of bootstrapping. We're not actually waiting for the observation to come. We're making some prediction, and observation to come, we can actually update our predictions for future outcomes on the fly as we go along. So, like a very relevant example for now with the weather, so say you want to know the weather on Saturday. So with the supervised case, we actually have to wait for Saturday, see what happens, and then maybe have a better prediction for the next Saturday. So yeah, so we have a model for the weather given some parameters and the observed weather in past days, and we want to know the future. So again, so in the supervised case, we have to actually wait to see what happens. In the TD case, so say say I want to know what happens on Saturday, so I can actually wait. So if I wait till Friday and I see what the weather is, I actually have a pretty good idea about what's going to happen on Saturday, and I don't actually have to wait till Saturday. I already have a prediction for Saturday, and this prediction can also give me some idea about what is expected on Monday, right? So that's the main idea. So yeah, so I'll go quickly over what Gal already mentioned. So there's lots of data that suggests that this kind of learning, the difference between what we expect and what we actually observed is this TD error, and this can be thought of some cost function in the sense that we're discussing now in Linda. And this kind of there's lots of data that is compatible with the theory of TD learning. And so this theory provides a signal that is appropriate for both predictions and optimizing future rewards. And the basic phenomenon is what Gal mentioned, that after training, the signals propagate back in time from the moment of the reward to the moment of some signal that predicts future rewards. Okay, so the basic findings is, when in early, we have some animal and we train it, and so in the early trials, reward is unexpected, and the dopamine cells respond to the actual reward. After training, they respond again to the predictor, not to the now expected reward. When we sometimes take the reward, we don't give it, like in the probabilistic case, the cells are inhibited, like I mentioned, and we have the precise timing of the prediction. And this is also seen in the behavior of the animal. Okay, so the shift from the reward to the conditional stimulus. Okay, so again, showing the same data that Gal showed, and all right, so the associated 5 different visual stimuli with 5 probabilistic delivery of rewards, 0 to 1, 0, quarter, half, and so on. The stimulus is held for 2 seconds and then disappears, like I already showed you. And so this is the main thing that we'll focus on for the rest of the slides. So basically, there's more, but for now, 2 main things, 2 main observations with the data, that one is consistent with TD theory and the other isn't. So one, TD theory suggests that this response at the time of the onset of a stimulus would scale with p. So when p is 1, it's largest, and p is 0, it's smallest. Later, we'll actually see the exact functional dependence of this activity on p. So this is consistent. But what isn't consistent, what's seemingly non-consistent, is that the activity in this time interval between the stimulus and the moment of expected reward, for potential reward, is that the activity should sum up to 0, but it doesn't. So maybe it's unclear now, but we'll see why fact that it doesn't sum up to zero it forms kind of a ramp shape, and like Gal said, because this ramp is most prominent in the case of p equals 1/2. Gal's paper, like I said, they suggested the reason is uncertainty because p equals 1/2 maximizes uncertainty, and the TD theory gives an alternative explanation. So if we look, if we compare, you have a question?
Question?
Yeah.
Can you go back? So you say that just in this kind of, because Gal said that there are two populations, right? The sustained activity and the phasic activity. So you say that the sustained activity is not compatible with the theory, and the phasic.
I'm, maybe I missed the terminology you're using, the sustained and phasic. I mean, yeah, I mean, you call this sustained and the rest is phasic, or?
That's the phasic and the other one. In the middle is the
The ramp
Oh,
is the
right.
sustained.
Okay. Okay. So yeah. So this part, whatever you want to call it, is compatible because it scales with p. Like we'll see it, why this should be expected. And what is inconsistent is this ramping effect that this doesn't sum up. There's a positive mean in this part. Okay. Okay, so if we compare trials which are rewarded or unrewarded, then in any probabilistic non-deterministic case, meaning p is different from 0, 1, there's going to be a prediction error. If the reward is delivered, then the error should be positive, right? Because the reward obtained is larger than what we expected, and conversely, if there's no reward, it should be negative. All right, so let's focus on the data that's inconsistent with the theory, or seemingly inconsistent. Why would we expect the average of these errors to be 0? Well, just by the definition of how we of the algorithm that we update our expectations. So if we want to minimize the error, right? That's how we update our model of the world. So if it's not 0, then we just this non-zero error would act as a plasticity signal, changing some kind of weight or model of the world, and then we should ultimately converge to a state of having no error. But this doesn't agree with the data where if we average over the rewarded and unrewarded trials, there is in fact mean positive activity, and this sustains even after months of training. So this is inconsistent. So then what the guys in Gal's paper suggested, they suggested uncertainty, and I'll skip over that because Gal talked about it. What they suggest in this paper is some kind of asymmetry between positive and negative errors. Okay. Why is this a good idea? Okay, so okay, so that's what they claim. We'll see in a few minutes why this is true. So asymmetry in the coding of positive and negative prediction errors leads us to see that, gives rise to the ramping effect that we see. And this also accounts for two other features of the signal, which is persistent activity, like we said, at the time of the potential reward, and weakening of the ramping signal if we have slow learning rate, and we'll see that towards the end. Okay, so okay, so the thing is that we have, why do we have this asymmetry? This is kind of a really straightforward idea, but I think, and I also guess that it's probably prominent and appears in maybe other contexts if we have some low baseline of activity and negative errors are um, shown with activity rate that is lower than this already low baseline activity, then we have a really small dynamic range for negative prediction errors, but we have a larger dynamic range for positive prediction errors. So there's a basic asymmetry in it. So if we had maybe a higher baseline activity rate, then maybe we wouldn't have this asymmetry. But once you're already starting with something that is already low, you can only go down to zero, right? There's no negative activity, then you have an asymmetry in the dynamic range of positive and negative errors. And so this just means that when we sum all these errors, the negative ones won't cancel out the positive ones because they have different magnitudes. Okay, so this would explain the overall positive mean activity. And the important point is that this is summed over trials, right? So, but in the in the brain, what actually happens, we're not averaging over trials, we're averaging over neurons. So this should pose a problem. Okay. Okay. What explains the ramping activity is what they call backpropagation of the prediction errors. Okay, so when we make trial after trial with the same animal, what we see is that the point of activity propagates back in time, like we mentioned. From the point of the potential reward to the point of, ultimately the point of the onset of the stimulus. And, okay, so, like this I mentioned. So, okay, maybe I should, okay, yeah. So, okay, this I think will clarify what I meant to say in words. So the x-axis is the time in a single trial, and the y-axis is different trials. These letters represent, like, R is reward and N is no reward, and this is in the case of p equals 1/2, and this is simulation based on their model. And what we see is that along the training, we see that y-axis on each subplot here is the activity of some 1 unit, and what we see is that as we go along trials, the activity propagates back in time towards from the point of potential reward to the point of the onset of the stimulus. Okay. So so this represents the activity of a single unit in their model, or a neuron. So each time step after the stimulus onset is consistently represented by the firing of 1 unit. All right. So the TD error, this delta, it's defined as okay. So let's see what all these letters mean. Okay. So R is the reward at time t, and W and X are the state and the weight vectors, respectively, of the unit at time t. So basically, this is the actual reward, and this is the expected reward, and the delta is just the difference between them. So if R equals the rest, then the error is 0, and actually what we do is we update the weights so that the error becomes 0. That's the yeah.
How does this equation fit into the?
I'm not sure I know how to explain that. I'm not sure. Okay, Nikki, can you? I don't know. I'm not sure. It's
I mean,
Okay.
if the delta was positive, then would the effect be later in time, and if it's negative, it will be smaller? Is that the connection?
No, no, the sign of the error doesn't determine the time of the activity.
I don't get it. Okay.
Um, or the this formula is this just is to illustrate the backpropagation, and they're
Yeah,
not
okay.
they're not directly related. Sorry. All right, and this is and this is the update rule of the weight. So we just take the the weight at the previous time step, and alpha is the learning rate, and we just multiply the error by the previous state of the weight. Um, okay.
Maybe 2 minutes.
Yeah. Yeah. 2 minutes. Okay. Okay. So. Um, All right. So okay. So if we have symmetric scaling, then between positive and negative, then we actually then we see the expected zero mean activity. But if we have asymmetric scaling, say d equals 1/6, which is kind of a realistic number that describes the ratio between the dynamic range of positive and negative, then we get this expected ramping effect. So, um, um, so, so that's this is how the model explains the data. Okay. 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 I'll go along so I can. Okay. Okay. I'll go to the end. Um, okay. I won't explain this formula, but it just illustrates that this is the mean error, and we see that and t is the number of trials, and we see that after when t grows to infinity, then we see that this expression is indeed proportional to the variance, because the p times 1 minus p is the variance of the Brunel variable, and we see that if d equals 1, then which is a symmetric case, then the error is indeed 0. But if it's between 0 and 1, then it will be proportional to the variance, like we said in the beginning. All right, so just to compare the two accounts, the paper that Gal presented, the uncertainty, so they would claim that the ramping is an within trial phenomenon according to the uncertainty, and the TD accounts would claim that the ramping effect is only apparent through averaging across multiple trials. Okay, just maybe this, and then I'll sum up. They also compared to see what happens when we have smaller, to see how the profile of the ramp changes when we have different learning rates. So alpha again is the learning rate in the update rule, and what we see is that the consistent activity here doesn't depend on the value of alpha; it's the same, but so the peak of the activity, but the width of the ramp depends on alpha. So alpha large means a fast learning rate, and small alpha means slow learning rate. Okay. these ramping effects can now be seen as kind of a signature of the TD phenomenon that up to the time of, of this paper was kind of elusive. Um, and the fact that they used, or the guys in Gall's paper used probabilistic conditioning is made made okay, never mind about that. Um, okay, sorry about that. Okay. So maybe I'll just give one, my own summary. Um, so I don't know. I was kind of more convinced. So this again, they're quoting the Gauss paper, and it seems that the TD account accounts for more phenomena and is kind of more consistent and a richer theory. So I don't know. I didn't go into depth with the uncertainty account, but um, I don't know. The TD theory seems to be kind of a more holistic theory and accounts for more phenomena. So um, I think it's kind of more convincing. What they in the end they offered to check in future studies is compare different. I mean compare what you mentioned in the beginning, like compare between intra and inter-trial averaging of the signal, and okay, never mind. I think I'll stop now. So, any questions? Yeah, go ahead.
They really talk about the dynamical process that gives rise to this ramp, right?
Yeah. The what?
The dynamical process, the
Mhm.
backpropagation.
Mhm.
And it seems quite straightforward to check if they took the data from
Yeah.
Gall's paper
Yeah.
and just try to check if the ramp builds up from from the end to the start, the beginning of the time, right? Because their simulation shows that the ramp kind of starts from the end and goes back to the prediction point, right? Or did I
Okay.
completely
Okay. Can
miss it?
you?
In the simulation that you
Yeah.
showed us
Mhm.
about the backpropagation, we saw that we have backpropagation.
This.
Things go, yes,
Okay.
from the reward to the stimulus, and the ramp kind of builds up, right? According to previous trials.
I mean, the ramp is only when this the ramp is only when you average many of these, and then when you sum up many of these. So you won't see a ramp in one. This is this is just one unit. The ramp is when.
So this doesn't mean
Yeah.
that the
So
ramp will.
so what we showed, what we saw before is each one of these thin lines, and when you sum them up, then you get the ramp effect.
No.
Okay.
Maybe I missed something. I don't know if you.
Okay. Yeah.
I feel like I have to stand up
Yeah.
in line. But they also see the ramp in single units. So that's, I don't know, how do they explain that? Or maybe it's just.
What did you
They
say?
see the ramp in single
They
units.
see the ramp also when they look at the single cell.
Yeah, but
Not
the
at
point
the
here is like
everything.
between trials and within trials, they see something that I don't really understand why that should be different. What you suggested is if you have asymmetric scaling, then the between trials will give you the ramp, but if you just average over all the neurons in one case, you wouldn't get it. And I don't see why that's not why is that so.
Can you say again?
So problem is that, as you say, you have a baseline, and with a negative prediction error, you can only go up to zero,
Right.
but with a positive one, you can go up to whatever you want.
Mhm.
So if you take one neuron and do it many, many times, and the neuron is kind of responding differently, like 6 times more for
Yeah.
the positive one than for the negative one, then when you take the average, you get something that is not zero. Okay. But
Right.
but if you take now the whole population of neurons, and all of the neurons are behaving in this way, why would the within tribe average? So you take only one tribe, and you take the average, why would it be different? Why wouldn't, why the sum would be, why
No,
cancel each other?
because that's where you introduce that D factor, where um, you you give more weight to the negative errors in order to compensate for this asymmetry. That's the thing.
Okay.
That's okay. Maybe that was one of the important points. So so
But there's also another point here that
Yeah.
which is like these neurons are well trained. I mean, why
Um,
would they? So there's
I
two
mean,
things that are kind of
what
very
you're
semantic.
measuring at different stages in the training. So you're measuring at the beginning and also after training. So what's the question?
The thing is that if you have all the time this asymmetry,
Mm-hmm.
then it's kind of how are you converging to how are you converging to steady state? It's like always you always have a positive signal.
Positive what?
You always have a signal. So why
Right.
would you converge, right?
Mm.
How do you get to a steady state? Unless it's an artifact, as you say, so it's not like it doesn't really signify
Uh,
a real error.
right. Yeah, you could consider this as kind of this representing the zero error, and any deviation from this would mean the but yeah. So it's kind of
So
steady, like I don't know. It's a steady state of positive activity. So that's okay, right? You got it.
no way.
Okay. I don't know. Mm-hmm.
Yeah, last right.
Okay, we're taking a break. Who's presenting after break? What is the
מה זה? אבל נכון, מכיוון אחרי המטרה,
אם הרב
אני
יסתכל.
מה קורה גם אם אין לך, כאילו גם אם הקיום שלך הוא לא אינפורמטיבי, כאילו, כן, יש מבחינה שהוא לא אינפורמטיבי, אז הוא לא ייכנס, כי הוא לא צריך אינפורמציה, וזהו, ואז בדיוק מה שהם מראים זה שבמקרה שיש כאילו רמפה כזאת, ב-0.5, אז יש שם באמת כאילו, אתה רואה תמונה שלא אומרת לך, אתה לא יכול להגיע למצב שהערך שלך יהיה 0, אתה אומר מה? אתה אומר
בחצי אתה אומר זה חסר
בחצי התמונה חסרת אינפורמציה, אז אתה פשוט ממשיך לחצי, זה מאוד
הבנתי, אז אתה אומר
ואז כאילו אתה יכול להתאים לדאטה של המקבל, באמת מצב שאין דרך, כאילו, אולי יש לי דילגים, אבל פה אין לי ממה ללמוד. בסדר, אני לא חושב שהם דווקא, כן, אני חושב שהנתיבי לרנינג לא צריך כאילו
לא, כי אז שוב, או לחלופין להרחיב את המודל שיסביר את עצמו בשפות, כאילו...
כן. אבל נראה לי שבאמת כשהכלי הוא חצי, אין לך פשוט איך ממה ללמוד.
כן, אפשר לדבר קצת על זה.
הערך של מצב,
לא מדברים על
שווה לערך של המצב
יכול להיות,
הקודם,
פספסתי, אבל
פלוס
לא.
את הדלתה.
סבבה.
אוקיי? עכשיו, אם הדלתה שלך היא מייצגת ציפייה, זאת אומרת שברגע שהציפייה שלך, אין לך ציפייה, הערך יישאר אותו ערך כמו שהוא היה קודם. אוקיי? עכשיו,
בעצם נכון עבור כל אחד מהמצבים.
עבור כל מצב, בדיוק, זה מצב, כל נקודה זה מצב, בדיוק. עכשיו, הדלתה שלך היא שווה לגמול המיידי שאתה מקבל, פלוס פלוס הערך של המצב הבא, פחות הערך של המצב הקודם. אז אתה יכול, יפה, בגלל זה אתה יכול לחשוב על זה כמו ציפייה, כאילו זה משהו שמייצג את מה שיקרה באמת, מה שיקרה לך באמת במצב הבא, אוקיי? פחות, כדאי לחשוב על זה דיאגנוס, מה שיקרה במצב הבא, פחות מה שקרה במצב הקודם.
מה שאתה חושב שיקרה או מה שבאמת יקרה?
מה שאתה חושב שיקרה. אוקיי? כי הערכים האלה, הvalue זה משהו שהוא פנימי.
כן.
אוקיי? אז המטרה, מה שיקרה בסוף, בהתכנסות, אוקיי, אז מה קורה? אז אתה זז בזמן. הערכים האלה זה 0, כי כל פעם העדכון פה, אתה מתחיל מ-0, הערכים עצמם, עם 0, 0, 0, 0, 0, אז זה 0 ועוד 0 במצב הזה, זה 0 ועוד 0 פחות 0, אז הערך שהמצב הזה יהיה 0. אתה זז למצב הבא, הערך הוא 0. עד שאתה מגיע למצב הזה, שהוא לפני הגמול שלך. אוקיי? ברגע שאתה מגיע למצב של לפני הגמול, מה יקרה? ה-R שלך יהיה שווה 1, הערך הקודם של המצב הזה הוא 0, ככה התחלנו אותו. אז עוד לא יודע כלום. אז זה 0, 0, ואז מה שקרה, שהדלטה של העדכון פה הוא שווה 1.
ואני מבין את זה שהוא יורד עם הזמן.
ואז מה קורה? ואז הערך של המצב שלפני זה, הוא נהיה eta, נכון?
באיטרציה הבאה,
בטרייל הבא, הערך הזה הוא eta, אבל אלה עדיין אפסים. זאת אומרת, אתה רואה שיש פה התקדמות back propagation של
הערך הזה הוא אתה לא הערך הקודם, הערך שלפניו, לא?
לא, זה מתעדכן לערך הזה, הגמול כאילו ניתן במעבר.
סבבה, ואז בטרייל הבא, אבל זה כן הולך אחורה?
בטרייל הבא, כבר הערך של זה יהיה eta.
והערך שלפניו?
והערך של הפניו, אז מה יקרה? הדלטה, אתה רוצה לעדכן את הערך הקודם, הדלטה יהיה R שווה 0, נכון? אז זה יהיה eta, פחות 0, ואז אתה תעשה עדכון של eta בריבוע.
הערך הלפני האחרון לפני 1 עדיין הוא eta?
לא, זה גם משתנה.
למה הוא ישתנה?
אוקיי, אז זה בוא נראה. איך זה יהיה גמול, עדיין אתה מקבל גמול נניח, אז זה 1, פלוס הערך של המצב הזה הוא 0, אנחנו לא מעדכנים אותו. אז זה 1 פחות eta, אז הדלטה שלך זה 1 פחות eta, אבל זה כפול, זה יהיה, אז הערך של המצב הזה זה יהיה eta פלוס eta כפול 1 מינוס eta, נראה לי.
שזה נגיד גדול מ-eta בריבוע.
זה בטוח גדול מ-eta בריבוע.
זה מה שאני אומר, נוצרת לך רמפה. נוצרת לך, בסופו של דבר...
זה התכוונת.
נוצרת לך רמפה לא במיצוע, אלא בטרייל עצמו. זה מה שאני אומר.
אבל לא, אין לך למה. אנחנו רושמים פה, נראה לי, במצב של התכנסות. אחרי שלקח כבר מיליון טריילים.
אבל היה את הסימולציה שקדירה. זה עובר, הפסיק הזה עובר אחורה.
נכון, אבל ה-TD,
הרמפה זה רק על מיצוע, ככה הם עושים.
אבל לפי הדינמיקה אמורה להיווצר רמפה, לא במיצוע. מה שהרקע לי פה זה רמפה.
נכון, זה לגמרי רמפה.
זה לגמרי רמפה, לא במיצוע, רמפה בסימולציה.
מה זה עכשיו? זה כאן? HDMI?
זה אני לא מבין.
למה אני לא מבין?
זה values.
אוקיי.
אנחנו אומרים שהערכים, מה שמיוצג במיצוי זה ה-TD error, זה הדלתות.
זה הדלתות, בסדר.
נראה לי, זה מה שהבנתי.
לא, רגע, מה שראינו שם, הגרפים האלה שעושים בקפגיישן, לא, זה ה-value, זה activation. זה לא רגע, נכון, מייצג את האירור, תכלס.
את האירור, אוקיי. ולמה האירור לפני 1 תהיה עכשיו 0 והאירור לפני זה יהיה אחרת?
אבל...
האירור, הדלתה לא אמרנו שהוא 0, אמרנו שהדלתה הוא...
נכון, הדלתה לא 0. בוא נחשוב על מה הערך של דלתה. אז בטרייל נגיד השלישי, שעכשיו אנחנו דנים בו, אז הדלתה הוא... צריך לחשוב... מה קורה במצב...
זאת אומרת שהשאלה שלי לא תקשיב טוב.
אני בטוח.
מה
קורה במצב...
לגבי האם יש רמפה
למה לא נוצרת? כאילו, למה...
ככה, זה ממש, כאילו, ממש אומרים את זה, שהרמפה היא רק בזה, ואני לא... כן, אני איתך. מה?
זה לא, כאילו זה הגיע לכאן, אבל זה לא מגיע.
אוקיי, אז זה שווה 1 מינוס דלתה. וזה, הדלתה של זה שווה ל-0 פלוס eta. נגיד שזה 0, זה eta.
איך קוראים לזה? שכפול צו או משהו כזה?
זה eta. זה יוצא שהדלתה זה eta, 1 מינוס eta, לא, זה אחת.
זה אחת, סבבה. תשמע, מה שיכול להיות זה שכעבור כמה זמן זה שואף לאפס, ואז הרמפה שלך היא כאילו...
לא, אני
זה
אשלח
בטוח
את
שואף לאפס בסוף.
זה נפתח
כי מתי
היום.
זה מתכנס? כשבממוצע הדלתא T שלך שווה אפס.
לא, זה כזה, אני מתכוון.
כן,
הרמפה, ברמפה זה שנה אחורה.
עכשיו, מה זה עבר? זה הדלתא או שזה הvalue?
זה הדלתא, הוא אומר שזה הדלתא.
ככה אני מבין, כאילו...
אז אם ככה יכול להיות שבצדדים, מתישהו זה מתנתק מהאחד ונהיה אפס. אתה מבין, מתי שאתה עובר מספיק שמאלה, אז כנכון הערכים האלה זה מה שילך שמאלה.
נכון.
ואז מצד ימין נוצר לך ערכים יותר ויותר קטנים ליד האחד, למרות שאני לא מבין למה הם יהיו יותר קטנים. הם אמורים להיות קרובים לאחד, הם לא אמורים להיות קרובים, כן עדיין מקבלים אינפוט.
הvalue הוא יהיה קרוב לאחת, אבל הדלתא ילך לאפס. כי לאט לאט הפרדיקשן error שלך יתאים בדיוק למה שהיה בערך הקודם. זה, תחשוב, זה ילך ליגדל, וזה יישאר אחת.
הvalue ילך ליגדל, הvalue ילך ליגדל.
עד שזה יגיע להיות אחת פחות אחת. ואז הדלתא של זה יהיה אפס. בגלל זה, כשאתה רואה בסוף עבור פי שווה אחת, אז כאילו יש לך פה ממש פי גדול, ופה כאילו לא אכפת לו, זה אפס.
אז כאילו הבאמפ שלך באמת הולך אחורנית בזמן.
כן.
לאורך זמן.
לאורך
טרייס. אבל בהתכנסות, אתה אמור לראות שאין בכלל באמפ. עבור כל אחד מהפרובביליטי.
לא,
אני חושב שרק עבור
לא,
השלב הראשון.
יהיה תמיד בזה, וזה אתה רואה כשאתה עושה סכימה, אתה רואה את ה...
לא, לא בהתחלה, אני מתכוון באמצע. בתווך,
בתווך לא.
כאילו, הבאמפ בעצם זה דינמיקה של התכנסות של ה-TDO.
כי בסוף ההתכנסות, הvalue של הכל אמור להיות אחת.
גם הבאתי כזה.
אוקיי.
אתה יודע אולי לחבר את זה? אני יודעת מה אני לא יכולתי שתכתב
זה
את זה.
תלמד את עכשיו איך
וואו.
שפיר.
רגע, אבל אמרנו להרצאה שלך, אנחנו עושים גם בהרצאה שלי.
מה
מה זה בעצם שלך, אבל האמת היא
פשוט, מה אפשר?
פשוט. רגע, אתה מבלבל אותי. אז טוב, מישהו יודע להעביר או שאני אשלח את זה?
רגע, למה להעביר?
כי חיברתי את זה ועדיין לא רואים את זה טוב. אני גדי הציע לי לשלוח את זה, משהו טכני. לחצתי המחשב נייד, זה באמת הגיע לפה, אתה רואה? אני חושב שזה הגיע לפה, אבל זה לא הגיע לכאן. טוב, מה, אני אשמור את זה ולעזור
לא,
פה?
לא, בוא פשוט נעביר את זה עם...
מה, עם מספרטים?
כן. למה להסתבך?
זה שהוא בתל אביב, יש לו מערכת. יש לו מערכת.
איפה הוא מחובר? Okay, so I'm a bit sick today. I think that I ingested in my body more grams of painkillers than food in the last 24 hours, so I hope my brain still functions. So if I say nonsense, please tell me. Okay, so I am going to talk about different subjects, the hippocampus. And the paper I'm presenting is called Internally Generated Reactivation of Single Neurons in Human Hippocampus During Free Recall. It was published in Science in 2008, and this work was done by Agar Gelber-Sagiv from Rafi Malach-Weizmann in collaboration with Yitzhak Fried. So already from the title, you can see that we are going to talk about single neuron in human hippocampus. So the recordings in this experiment and also in the experiment that preceded this experiment are from brain of live and conscious humans. So the subjects in this kind of experiment are patients with intractable epilepsy who underwent implantation of electrodes in order to localize the focus of the seizures. So these patients sit for a few days in the hospital and wait for seizures. So this is a great opportunity to record from the brain while they are performing all kinds of tasks. So what previous studies have shown is that neurons in the medial temporal lobe of humans respond in a highly specific manner to, first of all, to complex stimulus categories. For example, this is a paper from 2000 that showed that there are neurons in the medial temporal lobe that respond in a specific manner to different categories. For example, there is a neuron that is responding to faces, or to cars, or to food, or to famous people. So there is a selective response to categories. Another example is another finding is that they found that empty neurons respond to individual persons or landmarks. I'm sure all of you heard about the Holly Berry cell and Jennifer Aniston cell. So I had to bring it here. So the R neurons is responsive specifically to specific people. And what this is a paper from 2005, and what I wanted to see here that this is a neuron that is responding to Jennifer Aniston, and what's interesting here is that it does not respond to a specific picture of Jennifer Aniston, but to all kinds of different pictures. It means that the response is not to a certain physical feature of the stimulus, but to the concept of the stimulus. It's the concept of Jennifer Aniston. So the paper we are going to talk about today had two new goals in this field. So the first one, okay, Okay, so the first goal, note that in all the previous experiments, the stimuli were stationary stimuli. But when we're walking on the street, we don't see pictures of people. We see stimuli that keep on changing. So in this experiment, they wanted to test the response of neurons in the medial temporal lobe to non-stationary stimuli. So for this reason, they used video clips and showed the subject video clips. Another issue is that they, in all the previous experiments, the response was very short, and now they presented video clips to the subject, so we expect a much longer response. Another goal is they wanted to examine of this experiment is they wanted to examine the response of the neurons during free recall. So at the first step they showed the subject video clips, and then it's interesting to see what happens to the neurons that responded to the stimulus while the subjects are recalling this stimulus. So the two brain areas we are going to see recording from, and they focused in the paper, are the hippocampus and the entorhinal cortex, which I'm sure you're all familiar with. So the experiment was done as follows. As the first step, the subject was shown a series of short video clips. In the video clips, there were scenes of famous people, famous places, and known TV experience. Each video clip was lasted for 10 to 15 seconds. And clip was repeated in a random order in order to have many tries of the same video clip. After that, there was a short intervening task, and after that, after a few minutes, the subjects were asked to freely recall the video clips they saw, and they were told to verbally report the recalled video clip as soon as this video clip came to mind. And note that there was no external cue in this task, so the subject sat in a room and just had to freely recall, okay? So I think it's the most interesting part here. And now we can look at the results. So first we look at the result of the viewing session. So what we see here is a response of a specific neuron in the hippocampus of one of the subjects. And each square here is a different video clip, and the rows are different tries, and at the bottom we see the PST age for each video clip. And what we see here is that this neuron did not respond to most of the video clips that the subject showed, but it responds specifically to the symptom, a little bit also to the cycle. Okay, so we see a specific and a very significant response to one of the video clips, one or more of the video clips. And this is just an example. What they showed is that the majority of the recorded neurons showed a significant response to one or more of the video clips. So this result is consistent with the previous result from previous experiments. And I just want you to know that each video clip is lasting for a few seconds, even more than 10 seconds. So this is a very sustained response, and it's something that has not seen before. And now we can look at the result of the recall session. So what we see in this graph, the top graph shows the firing rate of the same neuron that we just saw, the neuron that responds specifically to the symptom. And this is the firing rate as a function of time. And here we see the sound amplitude of the subject. So the subject has to verbally report that he recalled a specific video clip. So when the amplitude increases, it means that the subject says something. So here we see that the number here represents the number of the video clip that he recalled. So for example, when you recall the video clip number 45, there is a slight increase in the firing rate. But what is interesting in this graph is that we see that for video clip number 46, which is, I guess you can guess, is the symptom, yes. So there is a very large increase in the firing rate of this cell. It means that this neuron that was, the same neuron that was activated while the subject was watching this video clip is activated when the subject is freely recalling the video clip. And if we zoom in to these few seconds, we can see a more interesting phenomena that this increase in the firing rate of the cell starts 1.5, you can see it here, but it's about 1.5 seconds before the verbal report, before the subject says the symptom. So I think this supports the idea that the claim that this neuron is involved in the retrieving of the memory of this video clip. Yes.
It seems like this cell responded also to Seinfeld, so could it be that it's just a humoristic cell? Like he also laughed when he said Simpsons, so maybe you think Simpsons is funnier than Seinfeld, but basically what it encodes is that just it's funny.
Funny thing. Okay, so it's a good question. I asked myself too because it's not even the only example that he laughing after the verbal report. I refer to this later, the D try, I have a slide on this, the D try to see whether there is a relation between different video clips that the cell responded to, but I think it might be, but not all the video clips are funny. So there's video clips of Saddam Hussein.
They didn't ask the subject if it was funnier.
They didn't refer to it, but they have many examples. So I don't have a real answer, but they don't have to. They don't know what is the relation. Okay. Okay. So this is just an example. Obviously they took all the neurons that were responsive, and they saw that most of them showed the same phenomena. So now we look. They show it the same result at the population level. So here they took all the responsive neurons from the hippocampus and entorhinal cortex. And when I say responsive neuron, I mean neurons that responded in a specific manner to one or more video clips. And they calculated the firing ratio between what is the firing rate of the cell while the subject was watching this cell preferred video clip, and compared it to the firing rate, the baseline firing rate of the same neuron. So here we see at the population level, it's average over all the responsive cells. We see that here time 0 is the time that the video clip started, and we see that indeed there is a very nice increase in the firing rate ratio at the population level. And the same here we see for the recall session. It is exactly the same analysis for the population level. Here time 0 is the time of the verbal report of the subject. So we see that there is an increase between 1 to 3 seconds before the verbal report of the subject. We see an increase in the firing rate, and all this at the population level. Yes.
Do you refer to the 8 seconds after the verbal?
This?
Yeah.
To just show it. I mean, I don't know what you can. Maybe the subject still remembers it or think about it.
Yeah, but there is decrease in them.
You specifically at 8.
Yeah.
No, they did not refer to it. I don't know what it means. If anyone has any.
Probably there's a lot of noise here. It's not very accurate.
That's the average or is it
Average.
So,
So,
Average.
no, it doesn't look like noise.
yes. It's average over 164. I don't think. Okay. So I want you to recall, maybe recall is not a good word. I want you to ask a question.
Do they do like control on that? Do they show any control?
They didn't
Control
show
in the
any
sense-
control to anything, but
No,
what control
control
do
of
you
the
think about?
analysis. So, for example, I mean, ratio is something it's a very sensitive measure because if you divide by something very small, you can get something very big. So, you know, so how would you control of this effect? I mean, what would you do in order to check whether this is real or not?
They did mention, I'm not sure if this is what you're referring to, but they did mention that these neurons, here it's the same neuron from above. So they did mention that the neuron here did not respond to during the recall of other video clips. They did say this.
But
And they also
I'm
said
sure-
that they took the baseline and specifically for each cell, they computed each baseline.
How would you check that?
What exactly do you want to check?
That what you have here is not an artifact of the analysis. Do you know what the standard way of doing that?
The permutation test?
For example, so some kind of bootstrapping. So for example, what you can do is like instead of taking time 0 as the time of the starting of the movie, take time 0 to be just random time. Okay, and then you run the same analysis on exactly the same data, but on random times, and then if you see something similar to that, then it means that something is wrong with your analysis or with your data, right? So something like that. Or for example, take one trial and another. So these are all kind of what's called shuffling or bootstrapping things that you can do in terms of this randomization. So you take the essential part, which is time 0 here, and you kind of play with it in order to see if this is real or just an artifact.
Okay. Okay, so I'll continue. So I want you to remember that the location of the electrodes in the brain of the patient was determined only by clinical considerations. These are patients with epilepsy. 2 minutes? But you were fasting.
4 minutes, right?
The location of the electrodes was determined only by clinical considerations. So they had recording from other brain areas. So this is a nice result they show. Here we see exactly the same analysis from before. The top graph are the hippocampus and the entorhinal cortex in separate. But what we see here is that we also have results from the amygdala and the parahippocampal gyrus. So what we see is that both in the amygdala and the parahippocampal gyrus during the viewing session, they are responsive neurons. We see exactly the same phenomena. Okay, but during the recall session, we see that the same neurons are much less active in the amygdala and in the parahippocampal gyrus.
every place they stuck an electrode, they saw that they...
No, 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 obviously in the... to
No.
any movie, yes,
Yeah.
yes, yes, yes.
Okay.
Yeah, yeah. It's, I think maybe, they showed the exact percentage of neurons that were responding to something. Okay, so, in the last,
One
yes?
more question. How can they explore all these areas in the brain? Because it's not a monkey that you can do it whatever you want to.
They didn't do what they wanted, I mean,
No, but you can...
It's in different patients that had to have the...
it's in different...
Yeah, yeah, yeah.
electrodes from...
Usually they put something between 6 to 12 electrodes in the brain of a patient, and this electrode has multiple types of records. So they want basically to sample as much as they can from the brain in order to do triangulation and find where the epilepsy starts. So they actually try to record from many places.
Okay, so just the last point I want to mention is, so the results I think are convincing, but and they are significant. But what is the meaning of the results? So I think it's clear to all of us, even by pure statistical considerations, that the neurons we saw must respond to many other video clips. They just weren't shown in this experiment. Okay, I don't think any of us think that there is a Jennifer Aniston cell. Okay, so what is the meaning? And this refers to Nick's question from the beginning. They tried to ask in the paper, and they didn't have an answer for it, but whether there is a relation between different video clips that a specific neuron is responding to. And I think they tried to imply that there is, and they showed all kinds of examples. Very, very fast, I'll go over them. The figures themselves are not important, but here, for example, at the top graph, they show an example for a neuron that the subject was shown 3 different episodes of Star Wars, but the neuron responds specifically to one episode. And this is a neuron that responds to 4 different scenes of Seinfeld. This is kind of interesting. They also showed here, it's a funny result I think, they showed a neuron that responds to 3 different seemingly unrelated episodes, but they said that they might have relation. The episodes are President Bush talking on Saddam Hussein. And the second one is Saddam Hussein itself. And the third one is the trial of the Oklahoma City bomber. And they said that there might be a war-like relation between these scenes. So I don't know. Maybe, as Neil said, there is a scene that's funny for the subject. I don't know. And here they also show 3 unrelated episodes. So I'll skip them. It's not very important. But what I want to mention is this example. I mean, I don't know what to say about them, but I think that what's really interesting here is that these are results from brains of live and conscious humans. So I think it's interesting to see this even to just, I don't know, think about it. I don't know what is the answer, but it's an interesting data. So I summarize. So we saw that a subset of neurons in the human hippocampus and entorhinal cortex exhibited highly reliable and specific responses during viewing of the video episodes. The same neurons showed an increased firing rate, again with free conscious recall before the verbal report. And it is not clear whether multiple clicks to which a neuron response may be related by some abstract association rule. And question mark. You already asked questions.
And did they say in the discussion, did they suggest to do a pre-recall before showing the clip?
But what would they, I mean, if you don't give any cues.
Pre-recall of a specific category. I don't know.
Just name 3 different.
Pre-recall of movies, I don't know.
First of all, no, they didn't. You're saying that maybe they expected they saw it before something?
Yeah, it could be that since like the priming effect or something, when you see something, so maybe these neurons are now somehow related to recent experiences.
Yeah, that might be, but the neuron did not respond during recall of other video clips. So
no, maybe they are specific. Maybe the neurons are specific to specific recent experiences. But it's interesting to see if these are long-term experiences also.
No, they didn't. I think that also during the viewing session, it kind of recall. I mean, topic that really interested me and they didn't refer to is whether um, the whether the store response in subject that, for example, did not know the symptom.
They didn't, what is it? The way they choose, I mean, from, I don't know, but how these experiments always work is they do an interview with the patient and they ask him like what kind of shows he likes and then they choose images that are related to stuff he likes to get a better chance to get a response. So
That
they
makes
can't.
sense. I didn't write it, but it makes sense.
So maybe not
Maybe it's obvious.
specifically here, but in other papers of this group that I saw, they usually do that because otherwise they can show you 1,000 pictures
Yeah.
and maybe not give the most.
Makes a lot of sense.
Okay, good. In the minute that Corey is setting up, I just want to mention two things. First of all, as Rotem said, it's a very interesting data, but it's also, you know that it's anecdotal, so in terms of very difficult to generalize from them. And also, the quality of the recordings are not at all close to what people record in monkeys or in mice, because these lectures are not kind of designed to do single units, and spike sorting is very complicated. The other thing is that there is a very similar paper from Rafi Malach group that is showing similar stuff with MRI, with fMRI. So it's actually, when you try to combine these two things together, it's very, very complicated because it's one neuron activated for Jennifer Aniston on one hand, and then in another paper it's a whole box cell that probably contains 10,000 neurons is activated, then it's becoming a bit difficult to interpret.
So, now that you're all dead and we don't have much time, we'll talk about something completely different, which is the directed differentiation of embryonic stem cells into motor neurons. Okay, so you can see that it's kind of a hardcore developmental paper. So I just want to make a very quick review of some of the stuff that we saw a few weeks ago. And we'll start with the two main axes of the nervous system, which is the rostral caudal axis. Okay, so the more rostral you are, the more you are closer to the front of the brain, and the more caudal you are, the farther you are down the spinal cord. And we also have the dorsal ventral axis. So your belly is ventral and your back is dorsal. Okay, and motor neurons, are they dorsal or ventral? No, motor neurons, they're ventral, okay? You can remember this because we do stuff with our hands and it's ventral, okay? So, motor neurons are ventral, and this is important to remember. And the second thing you have to take from this slide is the developmental stages of the spinal cord. So here you can see 4 developmental stages in the, is everything okay? Yes? Of the spinal cord, and 2 of the inductive signals that are related to the differentiation of different cells during these developmental stages. And we begin when the embryo only has 3 layers. So the outermost layer is called the ectoderm, and it starts to differentiate into a neuronal population. And these are called progenitor cells, or neural progenitor cells, and they are able to differentiate into any kind of cell in the nervous system. And so it's a homogeneous population. And upon the secretion of sonic hedgehog and BMPs, bone morphogenic protein, these are the 2 inductive signals we're talking about. This neural plate starts to fold, it creates the neural fold, and then it closes, it creates the neural tube. And if you go more caudally, you can see the spinal cord itself, and you can see that at the ventral part we have the motor neurons, like we just said, and at the dorsal part we have other kinds of neurons which are of no interest to us now. So if we know all this, if we have the knowledge about these inductive signals, it makes sense that we can use this knowledge to direct the differentiation of embryonic stem cells into a specific neural population. So the basic idea is that you can take embryonic stem cells and it then go through a series of steps and reach a specific population of cells that you want to get. Let's say motor neurons. And it's obvious that this could be very beneficial for clinical purposes because we could use it to cure or at least treat many different neurodegenerative diseases, like Parkinson, where we want to replace what? The cholinergic cells in the substantia nigra, or ALS, okay, where we want to replace motor neurons. So this is why we're talking about motor neurons here.
So just keep in mind that this is the whole goal, to take stem cells and make them differentiate into the specific subtype of cells that we're interested in. So what we're going to do is we'll review this paper in two parts. We have the first part where we see how they took stem cells in the lab and differentiated them into motor neurons. And the second part is to take these motor neurons and somehow transplanting them into embryos. Okay? Good. So almost all experiments you're going to see now were made on something called embryoid bodies. Okay? Or we'll call them for short EBs. And this is just a fancy name from 3D aggregates of stem cells. These are pluripotent stem cells. And they can yield microtissues very similar to native tissue structures. Okay, so it's kind of a natural culture of cells. And they can yield a great amount of cells, so they're good for clinical purposes. And what we're going to see now are some mouse EBs, mouse embryoid bodies, that were cultured for a few days, either in the presence or the absence of some added factors. And these added factors are supposed to be, from what we know from the literature, inductive signals in the development of the nervous system. Okay, so this is an embryoid body, and these are 8 different embryoid bodies. Now, as you can see here, what they wanted to see is the effect of RA, retinoic acid, on the differentiation of the spinal cord. Are you all with me now? Good. So we have two groups. We have the control group of embryoid bodies and the plus RA, plus retinoic acid. And we want to see if they express different markers. So here we have 4 different markers. And you don't really want to see many figures like this, one after the other, because you can't keep track of the names, like SOX1 or TX2 or NUN, right? So what I did was just replace these names with their meanings. So what we have is markers for neural progenitors, or markers for rostral identity, or spinal cord identity, or neurons. Okay? So now you can clearly see that in these two groups, we have two very different expressions of these markers. In the control group, without retinoic acid, we get many cells with rostral identity. We don't have any neural progenitors, almost at all. Okay, this edge is just an artifact of the microscope. And if we don't have neural progenitors, of course, we don't have neurons, and because they're rostral, they're not spinal cord cells. Good. But what happens when we add retinoic acid? Now we get many neural progenitors. Yes, Neil?
This is just a preset of the cells to become rostral?
Yes. The default is being rostral, and we have to add some added factors in order to make them become more caudal, a caudalizing effect. Okay? So when we add retinoic acid, we get many neural progenitors, and after a few days, in this case 3, you also get many neurons, and these neurons aren't rostral, but spinal. Okay? So just what you kind of mentioned right now is that exposure of embryoid bodies to retinoic acid promotes 2 different processes. Neuralization, okay, because it induces neural differentiation, and the expression of spinal positional markers, meaning caudalization. But what we want to see is if RA has a specific role in the caudalization effect. Okay, so what we need to do now is to find another way to induce neuralization that has nothing to do with retinoic acid. Okay, and they really did this, and they did it using something called PA6 cells. This is just a line of cells that is known to secrete inductive signals for neuralization. So what you can see here is we have markers in green for neurons and in red for rostral positional markers. And you can see that if we take these embryoid bodies and we culture them with PA6, what we get is we get some neurons and we get some rostral identity cells, and you can see that we have some yellow stuff here, right? So these yellow stuff are cells that express both kinds of markers, meaning they are neurons and they have a rostral identity. Now, if you add retinoic acid, you still get the neurons, okay, because you have the PA6. But now you mark for spinal identity, and again you get these yellow cells here and there, which means you get now neurons that have a spinal identity. So what we can conclude from here, is that indeed we can take embryoid bodies and we can induce them to become neural populations, and then we can subsequently make them spinal positional cells in order to make them spinal first axis, we know how to take cells and take them from becoming by default rostral and make them caudal. But we have another axis, which is the dorsoventral axis. Now, this is not an embryoid body. What do we see here? Right, the neural tube. Okay, so this is in vivo, a picture of a neural tube from a mouse embryo. And we can see that we have a different profile of expression of progenitor cell transcription factors along the dorsoventral axis. So you have in green interneurons progenitors. You can see them all along this axis. And in red, you have just a second, the motor neuron progenitors, and you can see they're only in the ventral part of the neural tube. Yes?
Technical question.
Mhm.
How do you make a picture like that in vivo?
I guess maybe it's not so vivo now. Great. So the question is, because we know we have sonic hedgehog somewhere around here, and we have these other stuff, these BMPs somewhere more dorsally, we have a gradient of these inductive signals, right? Yes. So what we want to see is, can we replicate this effect? Meaning, can we take different levels of sonic hedgehog, or in this case, sonic hedgehog agonist, and replicate this effect, meaning we'll get more motor neuron progenitors the more sonic hedgehog we put there. So here you can see many embryoid bodies, and you can see that the sonic hedgehog agonists that they use, these levels, they really control this shift in the profile of transcription factor expression. How can we see this? In these three left columns, what we see are markers for dorsal spinal interneurons progenitors. Okay, so they're dorsal, because we already know how to get dorsal cells. But these are spinal interneurons, which is not what we want. We want to get to motor neuron progenitors. Now, when we have no sonic hedgehog agonist at all, we get very bright pictures here, right? Meaning we have many interneurons that are spinal and dorsal. But what we want is to go farther down to where we add a lot of sonic hedgehog agonists, and then you can see that you have many bright pictures to the right, meaning that now we have an expression that's characteristic of motor neuron progenitors. And in the middle, you get something that's in between, which is what we would have expected if it's a gradient effect. This just summarizes it quantitatively, but we don't have time, and we don't, yes?
This is all done in embryonic stem cells of mice?
Yes.
Not humans?
Yes. It was done, we'll see if it was done in humans maybe 6 years later. Okay. So now, what did I want to show you here? Now we just want to make sure that we have not only the motor neuron progenitors, but also the motor neurons themselves, right? Because we don't want motor neuron progenitors, we want motor neurons that we can then transplant into different embryos. So what they showed here is that if they take retinoic acid and put the sonic hedgehog agonist, and they mark for motor neuron cells, they indeed get a lot of expression. And these are just controls to show that if you just add the sonic hedgehog agonist, you don't get these. If you just put retinoic acid and you put an anti-sonic hedgehog agonist, again, you don't get them. So this is just to make sure that we're doing it the best way we can. We have very simple steps that we can go through, and we can neuralize the cell, caudalize them, and then ventralize them. And what you can see on the right is that if they took just one cell out of these EPs, out of these embryoid bodies, what they marked here is, in green we have neurons, and in red we have the motor neuron marker, and you can see that the same cell indeed expresses both. So evidently the markers we're using, they're good markers. And if you lost me completely by now, this is all you have to remember. We have embryonic stem cells, and we can induce neural induction using PA6 cells. Then we get this rostral by default neural population. We can caudalize them, going from rostral to caudal using retinoic acid, and then we can ventralize them using sonic hedgehog. Okay, and now we're supposed to have motor neurons. Yes.
I should have asked this before, but so a neuron that is, either rostral or caudal or whatever has genetically.
Yes, it has different expression because it expresses different proteins.
So
you have markers for
neurons in the brain,
different positions.
They express different genes according to their position.
Hmm?
They all express, for example, neuron, right? So you can mark them as neurons, but pyramidal cells and motor neurons express
all the nervous system, but you have specific proteins that are expressed in different lobes or in different places along the spinal cord.
This is, for example, what we use when we want to, think of the genetics, we want to translate only the biologic neurons or only sensory neurons. So the biologic neurons are expressed in GAD because it's like a pre-processed GABA, and then you can see them to express in different places.
Yeah, it was just surprising that the location. I mean, it means that these different. Because I thought that it would be, I don't know, GABAergic neurons would be all across the brain, not
Right. So
really in.
GABA wouldn't actually tell you if it's rostral or caudal or
Yeah, but
If you have different functions in different positions, you can expect different
Yeah.
gene expression. So now that we know how to produce these motor neurons from the embryonic stem cells of mouse, we can take these and transplant them into embryos of another mouse. No. Thank you. No. We can put it in a cheek embryo. Okay. So this is what they did. They took these motor neuron enriched EBs. Okay. And more specifically, they were enriched embryoid bodies with motor neuron EGFP. So it has enhanced GFP, but only when this is a motor neuron. And they transplanted it into the spinal cord of cheek embryos. Okay, no question here. Great. And motor neurons were found. Here you can just see them. Okay, they're at the spinal cord where you would have expected. But the more interesting thing is, think about it. When you take these embryoid bodies and you transplant them into this poor cheek embryo, you can't really control the dorsoventral position, right? Because it's really a fine scale. But when you come back after a few days, you can see that the enhanced GFP, marking here motor neurons, is only at the ventral part of the spinal cord, but these mouse interneurons, they're all over. Okay, so great. So why? Thank you. Why cheek embryos?
Because they can.
Yeah. First, they can. Okay, it's quite easy to do the surgery there, and embryos of cheek are really the model for embryonic development. And second thing is, this is just an easy thing to do here. If you have a mouse and you transplant cells from a mouse into a cheek, then you can easily stain cells or mark cells that are from the mouse, right? Using antibodies, mouse antibodies in the cheek, so it's easier to see things also.
Did they say anything about what happens to these embryos when they?
No, they kill them all. Really, they kill them all.
Really?
Yes.
Why?
Okay. We'll talk about it later. But now what we want to see is, do they actually do their job? So they went to the embryos and they told them, okay, move, do something. But all the embryos were like, okay, they were embryos, they don't do stuff because they kill them. So what they wanted to see is if they have the three basic characteristics that you have to have in order to function well. Okay, so you can't see if they function, but you can see if they have everything they need to function. First of all, you can see here in a very beautiful picture that the motor neuron axons indeed follow the normal neural pathways. Okay, and here you can see that they exit through the ventral root and project to the limb. And moreover, when they project to the limb, they really they get to their target because they innervate the muscle itself at the end plate. And maybe the most important thing is that they know where to terminate because they terminate very close to clusters of acetylcholine receptors. Okay, which is what you would have expected from motor neurons. Good. What? Okay. So now to conclude, if we have knowledge from in vivo experiments about the pathways of developmental inductive signals, we can then exploit this knowledge in order to control in the lab extracellular inductive signals and take a population of embryonic stem cells and then make them differentiate into the specific kind of cell that we want. Now this is done without any genetic manipulation. So this is both kind of cheap and it's really, it goes through the process that happens in vivo. So this is really good. And if you want to think about the clinical applications that are possible for this kind of papers, then a science paper published only 6 years after this paper shows that induced pluripotent stem cells, okay, stem cells that were created from regular cells in a human being, in an adult, used to generate from patients with ALS to, they were shown to be able to differentiate into motor neurons. Okay, so this is very good because if you want to transplant cells from one person into the other, his immune system might attack these cells, right? But if you take in this very potent stem cells from the same person and make them become motor neurons, it's more probable that you could then transplant them back and cure this poor person. Do we have any time?
No.
No. Okay.
Question though. Question? So you all
Did
realize
they
that
say anything about why they don't see if these embryos develop into hypermotor activity chickens? No, I don't
Well,
know.
no, but why do they do this in embryos? Because embryos don't really have an immune system. So why did all these mouse cells didn't die? Because its immune system didn't attack it. So it's possible that if they would have waited later than what they call stage 27 with the development of the cell, then they would have died.
you all realize that this is science fiction.
Yeah, the paper wasn't really published. Just kidding. So, with that, thank you. Ja, wollen wir sprechen zusammen im Shiur?
לא, היא לא קבעה מתי זה. לא, היא גם לא אמרה מתי נבחר. לא, היא אמרה ששלחתם ביום שישי והיום נוכל ליום שישי הבא. אבל לא משנה, היא לא אמרה עדיין מתי נקבע.
צ'יק אמבריו פשוט היה בחורות שוות. אבל הן יהיו גם מחוברות. מה? הן יהיו גם מחובריות. צ'יק אמבריו מגנום.
תוסיף לכם עוד שניים.
לא חושב שהראו את זה יכול להגיד אם אני מעביר עכשיו למחשב נייד, אני יכול לבסס את הסאונד המחשב אני יכול לשלוח אותו עכשיו בווידאו מצלמה לא משתמשים בזה, אנחנו פשוט, יש כאן מצלמה, אנחנו לא משתמשים בזה. אני עושה כאן קו אודיו וקו VGA. זה מה שהם צריכים לעשות, זה הכל, זה מה שהם רוצים לעשות. לא רוצה שיתעסק בממשלה, כל מה שצריך זה באמת להתפרנס שם ולבדוק אם צלמת יש סטודנט הוא עושה לה סקייג'ול
אם אנחנו מחליטים זה יהיה בלי כלום, לא, אתה למקם את המצלמה אחר כך, זה קבל עד למעלה.
אבל למרות אתה לא צריך את זה.
לא, למרות לא, זה בסדר. למרות אין שום בעיה. אתה תדאג את זה למצלמה.
סבר למטה.